also like to thank the organizers for giving me the opportunity to speak here and present some of the results that we got started on this great section. I would like to motivate my talk the following three iconographic representations of molecular processes that do something for us in the broadest sense. Here on top you see icon for vision or for material light harvesting. At the heart of these two processes is actually a molecule called retinal. And the retinal absorbs light and it very selectively transfers this light energy into isomerization of one particular bond in the retinal chain. Instead, here in the lower part, I show you a nucleobase that usually sits here inside our DNA and codes genetic information. That molecule does something very different with the light energy. It selectively transfers this light energy into heat, not into any chemical change, fortunately. And you will find that there are a lot of cases where molecules do a very good selective job. And the question is, why are they so selective? The answer that theory indicates is it's really the topology of the photoexcited state. And I would like to walk you through this on the example here of these nucleobases. So, as I said, the nucleobases are really strong ultraviolet absorbers. And every time such a UV photon is absorbed, the nucleobases inside this DNA would love to make dimer formation. This dimer is really bad for reading genetic code. So our body has some repair mechanisms for that. But you find that even in the single nucleobase, there's a very efficient way of getting rid of this photon energy quickly out of the chemically active state. And this contributes most likely to the photoprotection of our genetic code. So here I want to explain to you how this really works in photoexcited states. On the left-hand side, there's a simple orbital scheme, pi orbital energy, n orbital energy, and pi star orbital energy of this nucleobase. In the ground state, pi orbital and the n orbital are doubly occupied. Now when you shine ultraviolet light on the molecule, you make a transition from the pi orbital to the pi star orbital. That is the chemically reactive state. So you want to get rid of this state before you actually can do any chemistry. How is that accomplished? Here's the potential energy surface, or I should say, better say, an artist's conception of the potential energy surface of this molecule. So pi star excitation is given here by this blue arrow. In the pi star state, the molecule wants to make chemistry, so the quicker you go out of this state, the better you protect the molecule. And so you can protect the molecule by actually transferring this wave packet to these regions of degeneracy with other states, with the n-pi star state, with the ground state. At these regions of degeneracy, the Born-Oppenheim approximation that you used for calculating these potential energy surfaces breaks down. When the Born-Oppenheim approximation breaks down, then you get coupling elements between the pi star state and these other states, and you can efficiently transfer population into lower-lying states, thereby protecting the molecule. So, it's not something new. People study this since many decades, and there are also great experimental results. And I want to show you one of them. This has been done by UV pump probe spectroscopy, and I explain you this scheme here on the right-hand side. First UV pump pulse actually excites the pi star state, now in the isolated molecule. Second time-delayed UV probe pulse ionizes the molecule. So you can now follow this wave packet via counting ions as a function of pump probe delay that is given on this side here. This is one of many experiments that comes from the group of Thomas Schulz, one of our collaborators, and you can see two prominent decay patterns of the ions here. One is 100 femtoseconds, really short. The other one is 7 picoseconds. So do you see anything from this spectrum, what the different processes are? Is this electronic decay or nuclear decay? Not really, so you need a simulation. So I want to show you one group of simulations that predict, by calculating potential energy surfaces, that this wave packet has free access to these regions of degeneracy that you also call conical intersections. And essentially, with potential energy considerations, you can predict that within 100, 200 femtoseconds, you're within this conical intersection regions, and then the molecule can protect itself by going out of the pi-pi star state. So here you would say that 100 femtoseconds is really the electronic process, the transition from pi-pi star to lower-lying states. The 7 picoseconds, what's that? Well, that could be the vibrational cooling in n-pi star and in the ground spectrum. Okay, you could be happy, but there are more theories around. And so there is another set of simulations where people predict a barrier between the Frank quantum point and this conical intersection regions. And the wave packet quickly relaxes towards the barrier in 100 femtoseconds. So here the 100 femtoseconds is a nuclear relaxation process. But then it's hindered by the barrier, and it takes 7 picoseconds until it can do the conical intersection passage. So here, nuclear is 100 femtoseconds, and electronics is 7 picoseconds. Also, these simulations are in agreement with the experiment. So that's a weird situation, and one that we're not very happy about, and it seems to be a very general situation. And I found a paper here by the group of Lischka that summarizes this trend very well. It is titled Electronically Excited State in Photodynamics, a Continuing Challenge. It's still a continuing challenge. And that may be, or mostly because of 2 reasons. First of all, doing ab initio potential energy calculations is a thing that we can do on the ground state. It gets harder on electronically excited states, and the accuracy of such barriers needs to be really high to predict the time constants in the correct way. The other complicated thing is this non-von Oppenheimer dynamics. The real simulations in a real dynamic context are pretty hard, and there are only 3 groups in the world that can correctly do that. So, what now? We need to do a couple of different experiments, I guess. Also, we need to do theory. But the experiments that I would suggest are the following. Usually, these decays here, whether it's electronic or nuclear, arise from the following fact. As the wave packet travels down in energy in this neutral manifold, you lose energy to actually ionize the molecule. You could ionize in the Frank quantum range, but you can't ionize anymore. For example, here, you can't ionize anymore in these different electronic states. So that problem needs to be overcome, and it can be overcome by using vacuum ultraviolet, extreme ultraviolet, and soft X-ray probes. So in this talk, I will concentrate on the soft X-ray probes, but towards the end I will also show the extreme ultraviolet probe. So in the following, remaining 20 or 25 minutes of the talk, I will briefly sketch how we can accomplish this soft X-ray probing by measuring Auger decay, then show you our experiments that were done at the LCLS, present a new picture of this photoprotection process, at least in the isolated molecule, and then give some outlook what's possible in the future. So as you see, we don't do any more ultraviolet probing, but our probe pulse now has a photon energy of 565 electron volts. So that's a real soft X-ray. Here I plotted these calculated orbitals of the orbitals that I showed you before, at least in the energies. And you can see again the pi, the n, and the pi star orbital. Now, very deep energetically inside the molecules are the 1s states. The 1s state of carbon at 280 electron volts, nitrogen 400 electron volts, and of oxygen 540 electron volts. This pulse has high enough energy to really ionize all of these guys, but in the weak field limit. So I'm talking about weak field physics, not strong field physics, in all of this. So we drill a core hole, and for example, we can drill it at this oxygen atom, or this oxygen atom over here, and we make a photoelectron, but we discard the photoelectron. We could do photoelectron spectroscopy. We choose to not do it because the FEL sources, at least the soft X-ray sources like SACLA and also LCLS, they are based on SAS, self-amplified spontaneous emission, and they're rather noisy. And so photoelectron spectroscopy is really hard to do. We choose instead to measure the kinetic energy of the Auger electrons. After about 10 femtoseconds, this core here decays. One valence electron falls down, the other one is emitted with excess kinetic energy, and we measure this kinetic energy, which is not correlated with the photon energy. Now we need to ask ourselves the question, which of these orbitals here contribute to the Auger process? In order to do that, we first of all measure the ground state Auger spectrum that is given here in magenta, and we do that with the synchrotron. And now we try to model that, and people that really model it, I don't do it, are Shungu Miyabi and Francesco Tarantelli. And so this black curve here is a simplified approach by Shungu Miyabi, where you actually see many of these lines. These lines here are channels, and they correspond to one valence electron falling down, another one being kicked out. We find by analyzing those lines that this n orbital here is a strong Auger contributor. Why? We said it before, because of the large spatial overlap. Yeah? This n orbital is essentially a lone pair orbital with p lobes at oxygen. When you now have an oxygen hole, you have a large spatial overlap, and therefore this is a large contributor. There are also some more low-lying orbitals below the pi that also have lone pair character, but essentially you can concentrate on these lone pair type orbitals. These make the calculation much easier. Yeah? So you could say that by going into the X-ray domain, by having a high degree of localization, you at least take some of this complexity of the spectroscopy away. So now the key question is, of course, how does the Auger spectrum change when we do the pi star excitation and when the relaxation processes occur? And this is really what I try to answer. So Artem already showed this, so I'll skip. But what I really want to show you is the interaction region. This is a vacuum chamber. Inside the vacuum chamber we have mounted an oven. The molecules are heated only mildly. 150 degrees is still a mild temperature for these molecules, although never reached in our body, fortunately. The molecules are then evaporated in the interaction zone, where first an ultraviolet pulse hits them, makes a pi star excitation. It's a weak ultraviolet pulse. And then, after some delay, a soft X-ray pulse hits the molecules. The soft X-ray pulse makes the Auger emission, and we funnel the Auger electrons through this hole here into a time of flight tube 2 meters long that has been contributed by our collaborators in Sweden, Buke, and Pfeiffer. So experiments with time delay at the LCLS are a little bit tricky. I think the people that do FEL experiments here can confirm that. Between the pump laser and the probe laser, you really have 1.5 kilometers spatial distance, so to speak. And it's really hard to lock the two, and we have observed during our experiments like runoffs of this time zero of the order of picoseconds. So it was possible to, after the fact, resort these data. This was only possible because people at LCLS do such a great job inventing single-shot delay characterization techniques. And I see that John is nodding. This is really a wonderful tool that we have done there, right? So large experimental facilities come with large groups. Artem already showed that. So this is also a group that is rather large, not the largest. It's not like Henry Chapman's collaborations. But there are about 30 people in this group, and many of them are inside PULSE or inside the LCLS. It's like, but there are also important contributions from Western Michigan, from the Max Born Institute, and from Perugia, and of course from Houston. So I want to single out Brian McFarland. He's here on the list. He was really the leading postdoc in this experiment. He now left us. He went to Los Alamos National Lab, and he got a director's fellowship. Really So now, the experiments here. You have already seen the Auger spectrum of the neutral molecule, not ionized nuclear ionization. That is this black curve. I showed that before. Now, what's the effect of UV radiation on the spectrum? If you shine UV light 2 picoseconds before the soft X-rays on the molecule, you get this blue curve. And you can see that the UV reduces Auger decay at a kinetic energy of 500 eV. It increases the Auger decay in the wings. So to make this better visible, we should do a different spectrum, and then we color code the different spectrum. So wherever you see now blue, that means you have less Auger decay induced by UV. Wherever you see red, that means you have more Auger decay induced by UV. And with that, I show you the full data set. So this data set took a couple of hours at the very end of our beam time. We had 5 days of beam time, and the first 4 days we essentially messed around. Looking for spatial overlap, looking for temporal overlap. It's getting better, but you still need to be aware that it takes a lot of effort to really assure that you hit your molecules with UV pulse and you probe the same molecules with your X-ray pulse. So, now, on the x-axis is the kinetic energy, on the y-axis is the time delay between UV pump and soft X-ray probe. Please don't misunderstand me, this is not an equidistant axis. We go out to 20 picoseconds here, 5 picoseconds, 2.6. After about 1.3 picoseconds, there's not a lot of transient change anymore. In the beginning here, we have a timing accuracy of 70 femtoseconds, and this is really where most of our important data comes from. So, now you see 3 spectral features. Feature number 1 here in this region, feature number 2, and feature number 3. And what they mean really becomes more prominent with the integration of the spectral regions. So in region number 1 and region number 2, you see prompt signals with the UV. Prompt rise, prompt decay that reflects about our time resolution. And then in region number 1, we get a decay of about 200 femtoseconds that is accompanied by a rise of 200 femtoseconds in region number 3. In region number 2, we stay approximately constant. So what does this prompt signal? Prompt means directly after the UV pulse hits the molecule, we get the signal. So we know that the only state that absorbs inside the molecule is the pi star state. That's the right state. So therefore these prompt signals must be associated with this pi star state. So I make this a little bit more prominent by doing a cut here shortly after time 0. And once more you can see the reduction in region number 2 and the increase in region number 1 in the difference plot. This is the signature of the pi star state in the difference plot. Now the question is, can we find out by theory if you have a good angle on this thing? So I show you on the top the calculated ground state Auger spectrum and the ground state and the Auger spectrum of the initially pi star excited state. You can see that the pi star Auger spectrum is blue shifted with respect to the ground state. When I now do the different spectrum, UV on minus UV off, I get flip, and indeed I can see that I get positive different signal in region number 1 and negative different signal in region number 2. And the most important thing in this experiment is this trace decays with 200 femtosecond time constants. That means we're out of the pi star state after 200 femtoseconds. Where do we go? A little bit later, it seems that region number 1 is flat, but now we have a reverse trend. We have positive signal in region number 3 and negative signal in region number 2. We'll try to find out what that is. It's not a relaxed pi star state of any kind, that's always blue shifted, but we find that the n pi star state calculation, given here in red, that really shifts the Auger spectrum towards lower kinetic energies compared to the ground state. So now when you do your different spectrum, UV excited minus UV not excited, you really get this modulation, negative, positive, that we see here. So, that essentially brings us to the following picture, summarized over here. We get, at electronic relaxation in 200 femtoseconds of the majority of the pi star state, at least as far as our signal to noise allows us. There could be some little bit trapped in here, it's hard to quantify. We at least see the n-pi star state appearing. Now you might ask yourself, Markus, what's going on with the ground state? Well, I have to disappoint you. We have really low sensitivity on the ground state. We did some calculations on this vibrationally hot ground state, and more or less these spectra look like the cold ground state. So Auger is not the ideal method to see the ground state relaxation happen. Might be other methods are way better suitable for that. Now, I want to point out that in the past there was a lot of thought about a 7 picosecond time scale for this photoprotection. And so people always looked inside the liquids. Nucleobases and liquids show photoprotection. Bernd Kohler and his group found that out on the sub-picosecond time scale, also a couple of hundred femtoseconds. And they thought, well, if the liquid and the gas phase are so different, it might be that the topology of these excited states that is really responsible, you know, the effect of a barrier, the effect of a gradient, it might be that the topology is very different. But now it seems that the topology might be very similar because we observe similar photoprotection principles. So now I essentially have a choice. How many more minutes do I have? Wow, that's a lot. So since Artem showed these beautiful results on the Auger spectroscopy and what really happens in fragmentation, I would like to jump a little bit in my talk and show you some of our results that I initially didn't want to show, but now I really want to show it. So, what you have seen so far is very weak UV excitation. Try to be in the single photon limit. But of course, it's fun to crank your lasers up since you can, right? It's like driving 95 miles per hour on the turnpike. So we drove 95 miles per hour and we cranked our UV laser up. And now I show you not different spectra, but really normal Auger spectra, not normalized, no difference. Here, in the different colors, is encoded the delay. So our zero delay is here, this one of the light blue curves. Yeah, before time zero, this is dark blue. And now, as we actually go up here, we encode time delay. And you can see that the Auger decay of around time zero and the Auger decay about 2 picoseconds after time zero is really remarkably different. It seems that out of this broad, undefined structure, you get some nice narrow lines coming out. And we ask ourselves, what the heck is going on? And we just cranked the UV laser up. We didn't know what we're doing. So only after the fact we knew what we're doing. Turns out that when you put into the molecule 3 times our UV energy, so 3 times 4.5 eV, you have enough energy to fragment the molecule. You go into an excited ionic state, and in this excited ionic state, you first lose isocyanic acid, and then you lose CO. I'm especially happy about the isocyanic acid because I'm from Gießen, and Gießen University is called the Liebig University. Liebig was, teaching in Gießen, and so this here was discovered by Liebig and Böhler for the first time, this isocyanic acid. So what really happens here in this fragmentation process? Let's go back to this. The small molecules really have very sharp features. If you look at isolated small molecules, isolated CO, isolated molecules of the type of isocyanic acid, you observe sharp lines. Large molecules are unstructured. You have seen the nuclear-based spectrum. Why is that like that? It's because of the density of states in the dicationic manifold. There are way more dicationic channels in larger molecules compared to small molecules. So what you really see here is the fragmentation after the UV excitation. And if you really analyze these line profiles, they get sharper and sharper and sharper. So if any of you theorists feel like you want to simulate that, please. We're looking for some theory about that. I mean, it's self-standing. The explanation, I think, is okay, but we would like to get some more handles on this. Unfortunately, nuclear CO doesn't fit that lines, so it must be some combination of isocyanic acid, NCO, and most likely also an excited electronic state. So here we really observe a line sharpening in the fragmentation process. In other molecules, you don't necessarily observe this line sharpening, as we have seen in Artem's talk. So now I come back to the original line of my talk, future opportunities. Where am I? Here. Okay. So in the X-ray world, we speak a lot about multidimensional spectroscopy, multipulse spectroscopy. And I want to show you our first attempt on some multipulse spectroscopy, not in the X-ray, but in the extreme ultraviolet. So here's an iconographic sketch that everybody who does multidimensional spectroscopy likes. You essentially take 3 pulses. 2 of those pulses make a transient grading inside your sample. And you take a 3rd pulse that you scatter off this transient grading, and so you get a 4th pulse out, which is the scattered first order of your transient grading. And so we were curious since many years if we could realize such a transient grading with optical pulses and then scatter an XUV pulse of the transient grading. And for many years we were not successful. But now I think we have some success with that because we took the right sample. So here is our experiment. We had a sample that is solid vanadium dioxide. I went into the material science side, the dark side of AMO science. I promise I'll come back again. But this vanadium dioxide, that seems to be pretty interesting because you can use either by temperature or by optical pulses a so-called insulator to metal transition. And the origins of this insulator to metal transition are not really clarified completely yet. So people are doing a lot of experiments on this. And now we created a transient grading on this vanadium dioxide with 800 nanometer pulses. So you can think now you have a substrate that is a grading structure, and stripes are metallic, other stripes are insulators. And now vanadium actually has an MH on the order of 30, 40 units, broadened out really, very much broader than the atomic vanadium. And now we scatter a harmonic continuum, so a multicolor pulse of this transient grading. This here is our 0th order that we block on the detector, and then we get some limited 1st order that is spectrally or spatially distant from the 0th order. And when we scan through our time delay, we actually see that 21.5 eV and 23 eV really are above the background. I admit it's a crappy signal to noise up to now. We're doing these experiments. This is just a couple of weeks old, so we continue to do that and hope that we get this machine to a point where we can actually also look at molecular systems. Lou, you're shaking your head. I know you're very demanding when it comes to experimental results. I hope when we see next time that we have some better results. But I really want to point out that this is already a big step forward for us, and this is really thanks to Jakob Grill and Emily Sussman who really pushed that forwards. And the sample that comes out of Stuart Parking's group at IBM is grown by John, and we have fruitful collaborations within the material division side of the SLAC. So with that, I think I am done, and I'm curious about your questions.
I have a technical question. I know most of the stories, but this time I'm very much worried thinking about the amount of UV excitation because you mentioned about intense UV case. So in your case, what is the spot size and what was the energy and how many fraction you actually excited?
Yes, I think we're on the order of 10-20% partial excitation. Measuring the spot size is not so easy because we used, that's also technical command, we used this Holy mirror. So a mirror with a hole in the middle, and we had partially our UV pulse overlapping with that hole. what we did to make sure that we really stay in the linear limit is we took an Auger spectrum of the photoexcited state and we swept through our UV energy. And so we saw saturation phenomena at a certain energy, and we tried to stay way below the saturation phenomena in the linear regime of the spectral change.
Set the molecule about 10%.
10-20%.
What about stability?
Spot size?
Stability, quantum stability. You are using UV, right?
Yes.
So it's not how much?
Well, I mean, okay, our important thing is in visibility that the pump spot is larger than the probe spot.
Yes.
Our pump spot was about 1.5 times larger than the probe spot, and when you looked at the camera, it seemed that we always had the probe spot inside the pump spot.
Okay. What is the size of the?
So the size is on the order of 100 microns for the pump spot.
What about 100 microns for UV and what about X-ray?
Smaller than that, factor 1.5 is on the order of 50, 70 microns, maybe. Hard to say because the microscope that we had was not really optimal for these kinds of cases. You should really do a blade scan of this, but then it's a matter of do you have enough time in order to invest in these blade scans.
I'm going to team up with Lars Madsen as a theorist. It looks to me if you UV excite into the shot, into the pipe excited state, you need, of course, some time to get to the avoided crossing region.
Yes.
And with some likelihood then, you follow that or you go diagonally through. So if you can adjust the XUV pulse length at a comparable time scale, and I don't know what your pulse length was.
75 seconds.
Okay, 75.
Actually, the soft X-ray pulse is shorter than that, probably on the order of 50 seconds.
Okay. But wouldn't you then, if you scan so to speak, over the pulse length, wouldn't you expect a big influence on what pathway you actually take? Because I was thinking that the avoided crossing that you follow there.
What do you mean? Should I scan the UV pulse length or the soft X-ray?
I want to follow the nuclear dynamics, your first step, the nuclear relaxation. You go up in the point-time content region.
Yes.
That wave packet will sit down.
Yes.
It will take it some time to reach the avoided crossing.
Yes.
And in order to go down,
Yeah.
to be dipole pumped down to the n-pi star state, the XUV pump is going to be on.
The UV pump.
The UV pump.
UV pump, no X-ray.
Right. So if you
No,
change the UV
actually,
time
I think
over that critical time interval,
yeah.
the time of your reaction should be very different.
So I might have caused some misunderstanding. The transition into the n-pi star or the hot ground states, these happen in the field-free case. Of course, you can try to control this interaction by the laser field, but the conical intersection manifold is extremely far away from the Frank quantum point. So this light energy is largely tuned from anything that is closer to the conical intersections. I do agree with you, it would be super interesting to actually shine, instead of playing with the length of this UV pulse, to shine another wavelength in there with some delay and also with variable length. And then you can really try to stimulate down into help photoprotection.
Auger's asked my question.
Oh, your question. Okay, John.
Markus, I'm just making sure I've understood this mechanism. So basically what you're detecting is your Auger spectrum, and from the Auger spectrum you can really identify the contributions from, say, n-pi star and other states in the molecule.
Yes.
So you're watching then the time dependence of those different contributions.
Yeah.
And you're deducing that there's a very fast electronic transfer into the n-pi star on about 200 femtoseconds times.
Yes.
Which sort of supports the school of thought that has a fast electronic decay followed by presumably slower vibrational
Yes.
or whatever.
Yes.
But this seems to be to do with the height of the barrier, the fact that the barrier is perhaps a little bit lower than...
And it doesn't take much to actually...
Exactly. So this measurement in thymine how general would it be for other molecules where there's photoprotection? Is this really always the case, or are you just claiming it's true in diamond?
Well, also in the other nuclear bases, it's of some relevance. There are various barriers predicted. They're not always between, let's say, the pi star and n pi star ground state, but there's also sigma states coming in there. But the general statement about the accuracy of the potential energy surfaces is true, and it has been summarized in this review by Hans Lischner and his group. We don't have yet enough of accuracy on the gradients, on the barriers, in order to really reproduce the timescales. Yeah, you might say, is it important to reproduce the timescales? I think for understanding what's really happening, it is important, especially if you, like Uwe suggested, try to control these processes. You want to shine in your control concept, right?
So I have a question about this spectra as well. So your peak, the experimental peak, is quite broader than the theoretical one.
Yes.
And I would argue it's shifted quite a bit also, especially
Yes.
given the energy distribution of these molecules, of the few EVs.
Yeah.
So can you say something about this debate of the potential energy surfaces, that is a barrier or not, based on this distribution, or?
So let me try to understand this question correctly. You would say that the Auger features, at least the ground state Auger features, does not completely reproduce the ground state spectrum, right?
I'm talking about the pi star. The peak,
Yeah.
507, 508 electron volts.
That one?
Yeah, is very narrow compared to the experimental peak.
Yes.
And also shifted.
Yes.
towards
Okay.
the right.
Yeah. I need to say something in defense of the people that do the theory. We knew when we came into the experiment that the experiment would be, relatively speaking, easy, because we would trick out the jittery pulses of the LCLS by measuring Auger. But we also knew that we would give our theory collaborators, Shungu Miyabi, Tut Martinez, and also Francesco Tarantelli, a real hard job. And so what you see here is not the best approximation that I'll show you. The best is actually this Green's function method that Francesco Tarantelli did. However, I want to be consistent. The Green's function method right now cannot do the electronically excited states. So what you see here is a simplified way. So it's called a configuration interaction single, where you only do single excitations, one particle, one hole excitations. And this has some error to that. So from these spectra, you cannot accurately reproduce the experimentally measured features. And that currently hinders us to really say, we know the branching ratio of n pi star and the ground state, because we cannot accurately really simulate that stuff. I also didn't do, as you see, I showed here the original spectra, not the different spectra. The different spectra look shifted and slightly different compared to that, especially with n pi star. But it's a step in the right direction.
I agree. That's why I wanted to bring it maybe as a challenge for the theoreticians, because
Yeah,
now we can generate a lot of quality.
Shungu tried to do this at a higher up initial level, and the problem is that you need to take dicalionic states in an energy interval, you see it here, from 520 to 480, 40 EVs, and the codes simply explode. There's too much density of states in there. So we simplified it by the CI single, and also by singling out the important orbitals that have like lone pair character. But it's certainly a big challenge, and it would be great if we could do that better. The important message is the pi star static decay. That is self-evident from the experiment already. But if you want to say now about branching ratio and that stuff, we either need to do a different experiment, and if members of the PRP are sitting here, we had one in. What?
Not only you would have an experiment.
I know. A lot of biologists have an experiment.
So a question for Yannick.
Yeah, directly to this theory part. I mean, is it known what a barrier relatively close to a contact intersection does to the whole picture of adiabatic potential curves? I would be a little bit nervous in terms of coupling of different channels if this makes sense at all
Yeah.
once you have a barrier plus a conical intersection. But I have no idea if somebody did that. Maybe you know. It's an interesting problem.
Todd did the complete dynamical simulation. Actually, he's the first and only one who did a complete dynamical simulation. And I cited this, it's a Hewlett paper from 2007 that you can actually read about. Of course, you got coupling between n pi star and pi pi star already close to the barrier region.
And does the normal kind of intuition still hold that the barrier get reflection and so on, and it's kind of independent? Because you spoke all the time independently of the barrier and conical intersection.
Yes.
And I was wondering if that still holds.
I think in that case here it still holds. It might be different in a different case because the energy spacing is still large enough compared to the n pi star state. So the geometries are sufficiently different. So you can kind of say, okay, first you need to go to the barrier, then you get small delta E, small energy spacing, and then you can mix. But in the barrier region, you cannot mix yet.
I guess I had some related questions, and I think maybe you've answered it already, but I was sort of wondering, does the vibrational energy in these various configurations really influence these calculations? I guess, or the reality. And I guess what you're saying, it seems like maybe there's enough approximations already in there that the vibrational excitation really wouldn't be the dominant thing that's causing the differences in these spectra. But
So on the pi pi star state, and on the n pi star state, we did that rather extensively. So we have a complete set of different geometries. That is one geometry here, that is a barrier, or at the barrier geometry. Then we have a transition state, like in the middle of the barrier, and those are all blue shifted. So whatever vibrational stuff you get in the pi pi star is always blue shifted, but you never get a red shift. So that allows us to really distinguish the pi pi star state decay from the n pi star state filling. It would be bad if vibrational excitation or different vibrational coordinate on the pi pi star state would give you a red shift. And then you cannot distinguish the electronic decay from vibration.
I guess I was wondering if that could help you sort of figure out where things are hanging around. If you could look at the shape of this and how it differs from what you see experimentally,
The
you can
best-
sort of tell where the stuff is.
The best fits the region in front of the barrier, but there also the wave packet might be the slowest before it speeds up again on the way to the conical intersections. Doesn't make really sense to look at the earlier spectrum at the conical intersection. You spend 0 time there.
Okay, maybe one last question from you, and then let's go to the problem.
Really quickly, only as a follow-up to your Michael. So if there is a barrier of significant height, part of the wave packet would be reflected and bounce around.
Yeah.
And if you then probed it with a second, say, 80 femtosecond or so UV, you might be able to see some oscillatory structure in
Well,
the electronic organ.
we had hoped we would see a coherent artifact, but the problem is you have so many other modes inside the molecule, and you got very quick LBR, and essentially, I think you would agree with me, you can give up on and see this way. But it would have been great.
You can go around and go off the other side.
Okay, thank you very much. I suggest that I still see some questions, but I suggest that we close the session here and go to the coffee break. And that we then meet again here in this room at 11. First, let's go.